Hello ladies and gentlemen, my name is Dylan Hamilton from C.A.R. Entertainment and welcome back to another AS1 Biology revision video. We're nearly done with cells, guys. 4 more videos: cell physiology we have today, then osmosis, and then mitosis, and then meiosis. So 4 more videos to go. It's starting to get tough now. This is probably the toughest section of it whenever you go into cell physiology, osmosis, and then mitosis, meiosis. But once we get through that, we're nearly done. We just have tissues and organs to do. And then we'll be done AS1, guys, and then we'll be taking a bit of a break, and then we'll bring you then the AS2 revision videos after half term in February. So after Valentine's Day we'll start to bring you out the AS2 revision videos. But let's get straight into this one because we're talking about cell physiology. So basically we're going to go straight in and go straight for it in transport factors. Basically the factors that are going to affect transport is going to be the size of molecules, as if you have smaller molecules, these will slip between the phospholipid molecules, while large particles will only be able to move in or out by cytosis. The polarity or non-polarity of substances also affect it, as the non-polar molecules will move through the phospholipid bilayer, whilst polar substances will have to move via proteins. The concentration of the substance on either side of the membrane also has an effect, as it will have to move from a high to low concentration by diffusion, and the movement against the concentration gradient is required, then active transport is needed. Now, to the transfer of relatively small molecules, you will use 4 different processes. You will use diffusion, active transport, osmosis, phagocytosis, and pinocytosis. We'll go into osmosis separately in a different video, and that will be quite in depth. But we're just going to start off with diffusion, and in particularly simple diffusion. So basically diffusion, net movement of a substance from where it is in a higher concentration to where it is in a lower concentration. That is an exact definition. You just learn that off and you will be sweet. But to actually go into simple diffusion, and actually specifically simple diffusion, basically it's just non-polar molecules passing through a membrane unaided. That's key, unaided. They're very small molecules, will pass through, but they have to be water-soluble substances, the water-soluble substances will be unable to pass through by simple diffusion, as the phospholipid bilayer is hydrophobic. So factors affecting diffusion, then there are 5 of them. They're pretty straightforward. We'll start off with the concentration gradient. The higher the concentration gradient, the faster you'll see the diffusion. It'll also be faster with a smaller molecule. It'll be faster at a higher temperature, obviously because more kinetic energy. You use your GCSE for that. The thickness of the exchange surface is also a factor, as the thinner the surface, then the more rapid the rate of diffusion. And then finally your surface area of the membrane, as you'll probably know, the greater the surface area, the faster you'll see your diffusion. So now moving on to the second type of diffusion, which is facilitated diffusion. This is supported by protein characters. So unlike simple diffusion, this is supported by the protein carriers. The protein carriers are basically just the binding sites matching the specific molecules, and this will assist the movement of molecules across the membrane. There are 2 types involved. You'll have your carrier proteins, which take in the diffusion molecule, changing the shape and releasing the molecule on the outside of the membrane. And you'll also have channel proteins, where the channels will be formed by proteins with a central pore, enabling charged particles to pass through. The ion channels will be permanently open, and some will be gated, and then some will also be open or closed, allowing the control of ion movement. Now going on to the third one that we're going to talk about, which is active transport. This is when molecules are moved across against the concentration gradient, so from a low concentration to a high concentration. It's the other way around. And energy ATP is required. This is provided by your mitochondria. So it basically just involves your protein carrier molecules with the substance being transported, binding to the carrier protein, that then changes the shape and releases the transported substance on the other side of the membrane, and these carriers will be specific to the particular type of molecule or ion, and these protein carriers use ATP as their energy. So basic differences you'll see between active transport and facilitated diffusion is that the substances are moved against the concentration gradient, and you will require energy, and also the mitochondria will supply the ATP energy that is needed. So skip it on, we're going to move on from osmosis, because obviously I said we're going to do a different video for that. We're going to go on to cytosis, and specifically we're going to do endocytosis. So cytosis is very important in transporting the larger molecules that are too big for protein carriers, and they also transport a big bulk of the smaller molecules too, believe it or not. So endocytosis is the movement of substances into the cell. Well, there's 2 types of endocytosis, which include phagocytosis and pinocytosis. Now don't get this confused with immunity, because phagocytosis in this respect is different. It's not going over like with phagocytes and engulfing pathogens, etc. But phagocytosis in this context is the uptake of solid particles into the cell within vacuoles, whilst pinocytosis is the uptake of solutes and large molecules into the cell within vesicles. The way I was taught this, phagocytosis is eating, pinocytosis is drinking. Okay, so solid particles eating, solutes drinking. So that's how you tell the difference. Endocytosis is when the cell membrane will invaginate around the substance entering the cell from the outside. This will then form your vesicle, pinocytosis, which will pinch off the inside membrane, and then the vesicle will take this into the cell with the fluid cell membrane allowing it to reform, closing the gap created by cytosis. So, exocytosis now. Exocytosis is the movement of substances out of the cell and not into the cell. You'll start off with a secretory vesicle being buttered off the Golgi apparatus, moving in fusion with the cell membrane, and the contents of the vesicle will be released outside the cell. The creation of a gap in the cell membrane will be followed by reforming of the membrane. This is a very important thing in secretion of many proteins from cells, including digestive enzymes and many hormones. Thank you for watching this revision video on cell physiology. I'll see you next time for our revision video on osmosis. I've been Dylan from C.A.R. Entertainment, and I'll see you next time.